have been able to join online at least, and because we were eagerly waiting for this lecture. And thank you so much, sir. I hope our students and we will be learning something new today. And now I'd like to request Dr. Maharshi Krishnadev, actually with whose initiative it all started. So I'd like to request Dr. Dev to introduce Professor Sengupta to all of us. Over to you, Maharshi.
Thank you so much. Yeah, thank you so much. It's really a genuine pleasure for me to introduce Professor Anupam Sengupta, and I would like to thank all the beautiful minds of the Department of Physics for giving me this privilege to introduce, to do this honor. So why it's a privilege? That takes me to his biodata. So Professor Sengupta is a FNR Attract fellow. So to explain what is the significance of this is FNR is basically the National Research Fund of Luxembourg. And they have an initiative called Attract. It's more of anti-brain drain sort of campaign to attract best of the minds to Luxembourg. And this Attract Fellowship basically is given to only 1 to 2, as I learned, only 1 to 2 fellows. And they get an award of almost 2 million euros for 5 years, which is almost equivalent to 20 or 18 crore for in Indian rupees. And it's very much equivalent to this Sovja Kovalevskaja award by the German Humboldt Fellowship. So almost on the similar lines. And so it's extremely prestigious fellowship, in other words. So and he heads the physics of living matter group at the University of Luxembourg, and he holds the bachelor and master degree from IIT Bombay in mechanical engineering, and then he moved to Germany to join a firm. And then he quickly realized that he's not meant for that, and he wanted to move back to academics. And he joined the Max Planck Institute for Dynamics and Self-Organization at Göttingen, where he received subsequently PhD with summa cum laude. So summa cum laude is in Germany, there is stratification of PhD degree. There's summa cum laude or magna cum laude. So summa cum laude is the top-notch students get this kind of, it's very difficult to get. I know personally one of my colleagues who got the summa cum laude, the only one out of hundreds I know graduating from Germany, and he has a Nature paper and also an HFSP fellowship like Anupam. So back to his biodata. So summa cum laude in PhD in condensed matter physics in 2013 for his work on liquid crystal microfluidics. Between 2014 and 2017, Anupam was a Human Frontier cross-disciplinary fellow, first starting at MIT, and then he moved to ETH Zurich, working on a range of problems on the physical ecology of microorganisms. Since 2018, Anupam is based in Luxembourg, where his multidisciplinary team combines material physics, microbiology, mathematical modeling, and machine learning to understand microbial response and adaptation to different dynamic settings from marine and freshwater ecosystems to the human gut and cancer environments. Currently, Anupam is the member of the Institute of Advanced Studies at the University of Luxembourg, and among other roles, serves as the director of the undergraduate physics studies of the University of Luxembourg. So I think we are in for a treat for the afternoon. And so it's all over to the organizers and the speaker.
Thank you, Maharshi. Thank you so much. And now, without further delay, we all are waiting to hear from Professor Sengupta. So, sir, if you can share your slides and start.
Sure. Okay. Is it visible?
Yes.
Okay. Yeah. Thank you, Maharshi. Thank you, Momita, and thank you also, Purba, if I am okay calling you by names today. I understand over the last couple of weeks, you put in a lot of effort to make this day happen. And as fate would have it, I'm not able to be present today in person, although I had the best intentions and I was really looking forward to be there in person, visit the campus, and also interact with you, the distinguished faculty members at Adamas University, and also get a chance to interact with the students. So it does seem it has to wait for maybe a future occasion. But nevertheless, I hope what I'll try to tell you today, I don't think it's new topically, but what might be interesting for all of us here is to see the potential connectivities between different fields, some of which actually happened because of my kind of hopping from one field to another. I started as a mechanical engineering student back in undergrad, then moved to physics and then into oceanography. And so now what my lab does is trying to understand microorganisms. In a bit, I'll tell you why microorganisms are important using principles of physics and engineering. So my most sincere thanks to all of you for this kind invitation and the kind introduction, Maharshi, you gave. It all started early this year, when Maharshi and me, we were at the same event organized by Human Frontiers. And we had a very nice chat at the time. And when I also got to know about the exciting work you are doing at Adamas. So I hope you will have something interesting to take from today's talk. And of course, feel free to interrupt at any time. I will try to keep an eye on the chat box, but sometimes maybe I might get a bit distracted into the talk. So if I miss out your chat messages, then feel free to ask the question again at the end of the talk. So what you see on the screen, let me see if I can get it to the pointer. Yeah, what you see on the screen, I think one thing you can directly pay attention to is the length scale. And what you start to see there is in the first image on the top left, the length scale is 100 microns. Below that, you have a length scale of 500 microns. Then top right, 1 millimeter, and bottom right, 2 microns. So essentially what you're seeing is these are different kinds of organisms, microorganisms. However, they are manifesting different spatiotemporal patterns and dynamics which span different length scales as well as time scales, as we will see. So on the top right, this is bacterial colony growing on a nutrient-rich layer, something we will discuss a lot during today's talk. Below, these are phytoplankton. These are photosynthetic algae which abound our oceans and lakes. They are responsible for 50% of the oxygen that we breathe. On top right, these are filamentous cyanobacteria. They are called Trichodesmium. They are one of the largest producers and let's say fixers of environmental nitrogen. So they're extremely important. And below, this is a photosynthetic bacteria. And the small little globules you see inside, these are actually light harvesting organelles that these cells have. So I understand a lot of you are working on photonics and also, let's say, material aspects of physics. And one of the things that I will try to showcase is how nature has developed strategies to harness these physical and material-based, let's say, attributes for enhancing biological fitness. So briefly about Luxembourg, I'm not sure how many of you actually got a chance to check where it's geographically located, but so it's a small country. The population is, let's say, a third of Kolkata. The country's population, it's about 6 lakh. It's surrounded between Belgium, Germany, and France. So our lab is located in Luxembourg City, which is in the southern part of the country. but it's quite centrally located if you consider the geography of Europe. So we are about 2 hours by train from Paris, some big cities, in Germany, Cologne, and then Zurich in Switzerland, Brussels. This is all between 2 to 3 hours of distance from Luxembourg. So the city is, it has an interesting, let's say, architecture. It's in 2 layers. It dates back to the history of the city. It used to defend. So this is the citadel is to sit on the top of the city, and the let's say the inhabitants would live on the lower layer of the city. However, now this is all spread out, so it's a beautiful hilly region. So if you happen to be in Europe, remember to pass by Luxembourg. So we are in the central campus in the city. It's called the City Campus. And just to briefly tell you, our university just turned 20 this year. So it was founded in 2003. And this is just, again, these rankings really don't mean much. One has to be careful in how to interpret these numbers. But just to give you an overview, the university in the last 20 years has made it into some of the, let's say, the top universities of the world. So currently it's ranking about within the top 300 worldwide. This is the Department of Physics. So we are about 300 colleagues and administrative staff and technical staff, and 20 full-time professors, and about 100 postdocs and close to 150 PhD students. And of course, we also have bachelor students and master students. So the research in the physics and material science department at Luxembourg, it's kind of divided into clusters. So essentially, there's a huge emphasis on sustainability. So there are very active fields of research in photovoltaics and sustainable energy. In fact, the department was founded on these photovoltaics and energy and hard condensed matter physics as the core themes. We have spectroscopy and functional materials, some of which I'm sure you are also working on a lot at Adamas. And there's a core strength of statistical physics and machine learning. A more recent addition is quantum science and technology. And we have the soft and living matter cluster. So our group kind of hovers between the soft and living matter cluster, the statistical physics and machine learning, and spectroscopy and functional materials. So what exactly is today's topic? Microbial active matter. So microbial, essentially, it's the adjective of the word microbe. And microbe, any organism which is of micrometer scale can be generalized as a microorganism. So for today's talk, it would essentially refer different kinds of bacteria, different kinds of algae, phytoplankton. And why are these called active matter? So if you look at any material in nature, they can be categorized broadly into active matter and passive matter. So passive matter is what we typically encounter in most of our daily activities, and also what we study it during our high school, undergraduate studies, and so on. And the reason this is passive matter is because if you look at the guiding laws which describe the phenomenology exhibited by this matter, it's essentially, fundamentally, it's equilibrium thermodynamics. But when we break this equilibrium thermodynamics, you enter the regime of non-equilibrium thermodynamics. And that's what is essential component of active matter. And why is that? Because if you take any example, for example, if you take bacteria, bacteria takes certain molecules, generates energy, and then it essentially executes work and does some function. And so is the case with any kind of biological matter, be it a simple eukaryotic cell, be it complex tissues, be it organelles, be it organoids. These are then higher levels of biological complexity. But one key thing, what remains consistent among different classes of biological matter is that these are all out of thermodynamic equilibrium. So they consume energy, they exhibit, they perform some tasks, they can self-organize, and there is a constant feedback between the organisms and the environment. So they have the capacity to tune their environment, and that modified environment feeds back into the behavior and response of these organisms. Finally, the last point which I would like to briefly mention, what characterizes active matter is their property of demonstrating emergent phenomena. So emergence, you might know from dynamical systems, this is essentially a key facet of active matter. So if you look at a single cell, what it can do as a single cell is much different than what cells in collective ensemble can execute. So essentially, in simple arithmetic terms, the sum is greater than the two, essentially. So if you have 1 plus 1, the moment multiple microorganisms come together, they can do much more than what each of these individual organisms can do as let's say, a linear superposition. Okay. So now the question always is, do we have a framework to capture these interrelations? These interrelations as in relation between cell and cell, the cell with the environment with the cell. And so it so turned out, so there is this, again, I'm relying a lot on material phases, passive material phases in describing these systems. And one very prolific, let's say, material phase, which is found ubiquitously in biological system, is the material phase of liquid crystals. So I believe we all are aware of liquid crystal displays. You have it in your drawing room, you have it on your desktop. So these are essentially, it's an intermediate phase of material which combines the fluidity of isotropic liquids, but the crystallinity of crystalline solids. And what is interesting about liquid crystals is that they can flow, as well as they have some long-range order. And this long-range order could be orientational order or positional order. And it turns out, depending on which biological system we take, any biological system would fit into this whole spectrum between isotropic liquid and crystalline solid. So hence, liquid crystal offers a very nice framework to understand biology and, let's say, the emerging interrelations with the environment and so on. So when we describe liquid crystals, one of the key parameters is the so-called order parameter. So an isotropic liquid has no order. A perfectly ordered crystalline solid has a perfect order parameter of 1. And in between, you can have nematic order, synectic order, and so on. Now, if you look at different kinds of biological systems, this is just, this is by no means exhaustive list, but what you start to see, some of the very key molecules which are implicated in origin of life. It could be RNA, nucleic acids, diverse mucous systems, amyloid fibrils, food materials. They all have different liquid crystalline attributes. Furthermore, if you look at the mucus in your gut lining, or if you just look at a bacterial colony growing, the way these systems self-organize, they have very strong signatures of liquid crystallinity in it. So at the time, a few years back, this whole field of active matter, but active matter based on liquid crystals started to emerge. And our, let's say, 2 cents into this field was to understand different microbial systems, which we call microbial active matter, in the frame of liquid crystals and liquid crystal dynamics. And what it does is, by following this framework, you can really cover diverse microbial systems, from wound healing, tumors, cancer, microbiome, as it covers different scales of heterogeneity in both length and time space. So briefly, why are liquid crystals interesting, especially in the context of biology? Whenever we look at any biological system, because these are active systems, there is a constant flow of information, energy, and material. So either or, but generally there is always a dynamic element in it. And because there are also structural aspects, these flow, whether of energy, material, or information, this is coupled to the internal structure of the system. And this is something I was working on during my PhD, where we found that these systems are intrinsically coupled. So there is a coupling between viscous forces, elastic forces, and surface phenomena or surface forces which are present in the system. And broadly, you can start to describe them in terms of different non-dimensional numbers, one of which is Reynolds number, which is commonly also in fluid mechanics. It's a well-known Reynolds number. The other not so used non-dimensional number is the Ericsson number, which essentially compares viscous effects with elastic effects. And one can, of course, express Reynolds number in terms of Ericsson numbers. So these K, rho, and eta, these are all material properties of let's say, either the active liquid crystal system or the liquid crystal system. And you start to see that you can really get low Reynolds numbers where the Ericsson numbers are very high. And that has some very interesting consequences, which I'll show briefly. The other aspect is, whenever you have these anisotropic system, anisotropic meaning out of isotropy, if you apply an external field, for this particular case, I'm just showing hydrodynamic field, the emergent or the resulting behavior that will be recorded, that depends on the structural orientation or structural state of the system. For instance, way back in 1946, Miosovich was studying how the apparent viscosity of liquid crystal materials are dependent on the direction of the overall liquid crystal molecule. So these bluish rods that you see, this is the orientation of liquid crystal molecules. It's also called the director field. And these arrows, these indicate a Poiseuille type flow. So this is a pipe flow. And what you start to see is there are different viscosity coefficients. In an isotropic fluid like water, we would never expect different viscosity coefficients depending upon how water molecules are oriented. But in liquid crystals, it matters, right? But this is just an example that hydrodynamic fields are determined by molecular orientation. It could be even any other field, you name it, optical field, electric field, magnetic field. It's just like essentially you are bringing in the whole theory and physics of crystalline materials, but now into a crystal which can flow. And this has some very interesting also optical aspects. For example, these are some cholesteric liquid crystal phases. Cholesterol, which we are all aware of, was one of the first liquid crystal which was discovered. And when you study these under polarized optical microscopy, you start to see there are these beautiful oily streak patterns which start to emerge in these systems. These are lyotropic phases. So these are liquid crystal phases where it's not just temperature which determines their liquid crystallinity, but also the concentration of the surfactant molecules in a liquid. So these are one of the most relevant, biologically relevant liquid crystals you can think of. And there are different possible, let's say, means of harnessing these liquid crystal-based properties in assessing biological systems. For example, there's this fluorescent dipole orientation imaging, where by shining polarized light and the signal that you can get a very accurate measure of the internal structural order in biological systems. The other complementary technique is the level-free polarimetric imaging, where you do not need any essentially dye molecules, but simply by shining light, provided that the structural features are comparable to the wavelength of the light which is being incident, you start to get a lot of critical and important structural details of these systems. So these are some examples which are closer to bacterial systems, something that we very extensively work on. You might have heard of the word biofilms. So biofilms are one of the most, let's say, challenging structures to tackle in today's world when it comes to infection, when it comes to antibiotic resistance, when it comes to wound healing, and also when it comes to the gut microbiome. So practically any surface on Earth can be a potential substrate for biofilm formation. And interestingly, when these biofilms form, they also have some birefringence properties. These birefringence properties emerge because of the biopolymers which are present, which are exuded by these bacterial cells. And when you shine light, because these polymers have a certain radius of gyration, that imparts it some level of anisotropy. So naturally, when you now shine in polarized light, and you can really do these experiments very easily in lab. You just need 2 polarization foils and then just put them at 90 degrees with each other. You put the sample in between, and you start to see some very interesting optical phenomena purely in biological systems. This is again a very interesting system where this is P. aeruginosa. This is one of the most potent infectious bacterial species that we have. And so people have really used these optical birefringence, again, based purely on the liquid crystal physics, to identify different stages of biofilm growth, their virulence meaning how harmful they are, and their different physiological states and developmental states. So these are experiments one can really do easily in lab by precisely controlling different parameters. One can get into some very interesting physics and biophysics of these systems. So at the time, the other aspect that I was working on is that because I don't think it will come as a surprise whenever we have an ordered system, disordered singularities sneak into this ordered system, and these are called topological defects. This is something that you might know from grain boundaries, from Schottky defects, from, let's say, integer defects, topological defects. There are different kinds of defects. I would not go into details, but again, such defects are ubiquitous also in liquid crystal systems. The liquid crystal display industry does not like it because you want your liquid crystal display to be defect-free and smooth and clear. So what we did at the time was we benefited from microfluidics, and we used hydrodynamics to tune and tame these defects. So we could create these defects at will, and then essentially give rise to different phenomena, but also use them as, let's say, soft rails that could guide colloidal particles on these defect structures and defect-induced flows. So when there are a lot of defects, you can really also induce mixing at microscale, which is a major challenge challenge, especially in drugs and medical industry, because mixing is a key aspect, but one can leverage defects to induce mixing. Defects can couple with defects in other fields. So we saw in some of the works, we showed that defects in material field can couple with defects in hydrodynamic field. So these are some of the work. And finally, more recently, we are looking into defects in biologically relevant liquid crystals. For example, liquid crystals that phases you find in our gut, in the mucus layers at different parts of the body and different bodily fluids. And these are typically lyotropic phases, which are also responsive to its environments. And these are some of the recent works we have done over the last couple of years. Now, topological defects, these are hotspots of physical and chemical activity. So this is known. And the question thus remained, now that there is this interesting analogy between these passive liquid crystals and active biological liquid crystals, do topological defects have any biological function? And then with this aim, we started our research in Luxembourg. And one of the, let's say, very interesting books I had come across towards the end of my doctoral studies was Liquid Crystals in Biological Structures. So it's a very anecdotal book. You can really have a very nice read without even getting into too much of biological details. But this book really showcases some very interesting examples from human body where liquid crystallinity is evident. So broadly, also motivated from how this book was approaching some of these aspects, what we try to do in our lab is essentially, for any of the biological systems we're working on, we look at the geometry, order, and topology. We quantify them, we characterize them, we heavily use machine learning and image processing to access these details. Then we look into the emergent properties, and finally try to do molecular and, let's say, biological analysis to connect these to potential biological functions. So essentially, we are trying to look into how topology could have biological implications in these systems. So broadly, we look at these systems in different scales. It could be at the population scale, it could be at the community scale, it could be at the species scale. So community meaning you have a combination of different populations coexisting, and there is an interaction between these different populations. Population scale meaning you have a single species but multiple entities, and species meaning you have just, let's say, a couple of cells. You're either looking at single cell level or at subcellular level. And these are some different examples from mucus layers to food systems. This is essentially in some kind of a cheese. You have a huge presence of certain specific bacterial types. This is degrading biological matter. These moving dots that you see, these are bacterial cells, and these are some algae exhibiting some bioconvective motion. So microbes can actively tailor their environment. They can customize their habitats to regulate their fitness. And no wonder under this current climate, let's say, concerns, there has been a growing voice, especially among researchers working in the field of microbial systems, is that we need to protect these microorganisms, because the diversity in a microbial ecological context, this is extremely important for the resilience of the system. So what has happened with the specific anthropogenic stresses, be it change in the pH, be it rising temperature, be it the rising, let's say, turbulence in the ocean, these are specifically targeting certain microbial types, but essentially they are breaking down these microbial network which has over eons existed. And these networks confer resilience to the systems. And in absence of that, they push the systems to critical transition. So already in many ecological settings, the systems have already reached their tipping point, so-called, And this is also very interesting that such tipping points, the physics of these systems are also relevant for what is happening inside our human gut, or for example, what is happening inside the microbes which inhabit our lung systems, or inside the our mucus linings inside our body. So by knowing this physics, there is a lot we can contribute to address some of the current and contemporary concerns, especially in health and climate sciences. So how do we do these studies? So we have a bottom-up approach. So typically, something we take a lot of, let's say, benefit of is microfluidics. And generally within the microfluidic system, we combine it with different tools and methods to create and mimic environmental conditions which are relevant for the microbes. For example, we can create concentration gradients. So microbes typically inhabit environments which are experiencing different gradients. At the same time, we can measure different phenotypic traits of the cells, how fast they are swimming, what is their body size, how it's evolving over time. And then we can do imaging in 3 dimensions, so to get a complete picture. And in doing so, we are able to essentially understand both spatial and temporal evolution of these systems under very carefully designed environmental conditions and conditions which mimic, let's say, perturbed conditions due to climate change or due to unhealthy food habits, for example, if it comes to our gut experiments and so on. And we generate these using different combinations of soft lithography, 3D printing, and a generous dose of automation, and the quantification is optical and also spectroscopy. And once we have these details, then depending on the question we ask, we also combine this with molecular studies so that we have a full picture and potential biological consequences. So when we combine this, is there a question? Sorry. Okay. So when we combine these, there are also some interesting byproducts of this research. And one of the things is how microbes are responding to the current climate change. It's a very interesting field of research because it gives us insights into how nature responds to disturbances in its environment, right? And that gives us avenues to devise responsive biomaterials or smart biomaterials. But on the other hand, this also gives us potential ways to tackle ecological concerns and evolutionary concerns. So these are also some of the more translational aspects of research that we are interested in. So these are some of the projects that my lab is currently involved in. Microbiomes in One Health, and then there is this project on cancer microbiome, which I'll briefly talk about. And broadly, it fits this physics mid biology initiative of the University of Luxembourg. So this is very briefly a quick snapshot of some of our recent works, where we have been trying to understand how single cell bacteria self-organized as they grow, but also what is the role of noise in this system. By noise, I mean, in biology, unfortunately, we cannot control the properties as precisely as we can do in, let's say, material sciences, right? Because this is biology, this is nature. There is always an intrinsic variability between cell to cell. So one of the recent focus of our research is knowing that there is a variability. If we characterize this variability, how can we understand the population's ability to survive in different, let's say, stressful environments? The other key focus area in our lab is understanding photosynthetic algae, how they respond to turbulence, hydrodynamic turbulence, but also how they respond to different nutrient limitations and light conditions, because due to the current climatic change, all of these factors are getting disturbed compared to how these used to be for millions of years for these cells. So these are some of the earliest colonizers of our planet, and what they are encountering now is something dramatic, something they haven't seen, and that's why we are trying to systematically understand these different effects. And finally, we have a series of studies where we are looking at interrelations between bacteria and algae, because if you really look into natural systems, they coexist. So bacteria and algae, they are typically part of the same ecosystem, and impact on one influences the other and vice versa. So specifically for these bacterial growth, what we typically look into are these three primary axes, where we tune the environmental complexity through microfluidics, where we can tune what kind of chemicals they are facing, what kind of surface properties they are encountering. The other axis is the kind of bacteria and their phenotypic traits. It could be the shape, it could be the motility. And finally, moving from a single bacterial type, what happens when we look at the community structure. And this community structure is very interesting, especially when we come to the some of the specific cancer types. So we are working on the specific patient-derived organoid. This is called, this cancer is called colorectal cancer. So this is a major cancer which affects primarily women in Europe. And what has been shown is that this cancer is associated with dysbiotic microbes. So microbes which have gone rogue, microbes which were meant to do good in the gut, but somehow due to lifestyle choices, due to food habits, these microbes are no more beneficial, but they are directly promoting the progression of this cancer. And one of the things we are trying to understand is through very careful both spectroscopic and optical measurements, trying to quantify what kind of forces these microbes exert on the cancer and the tumor cells, because these forces are very interestingly, they determine the progression of cancer. So they have the extracellular matrix and these micro forces, Newton forces are enough to change the fate of cancer cells as they move forward. So they still work in progress, but it's a very interesting domain of research where you are essentially looking at two active systems. The tumor itself is an active system, but then on top you have bacterial consortia which are influencing the growth and progression of the tumors. Now, this is how a bacterial colony looks like in an intermediate growth stage. So if you look at the length scale, so this is about 5 micron. And what is interesting is if you start to carefully look at, you start to look at these, see these topological defects. And these defects are all over, and as the colonies grow, the number of defects keeps on increasing. And this is something not one expects in a passive system, because topological defects cost energy. And in a passive system always tries to minimize its topological defects. But in this case, the number of topological defects goes on. And this is because there is a constant influx input of energy into the system. And this is able to keep the defects and amplify the number of defects. So this is a step-by-step demonstration of how the defects are increasing. G stands for generation, so generation 1, 5, 6, and so on. And what you start to see as the number of generation increases, overall, you have the number of defects exponentially increases. what also happens simultaneously is there are flows which start to appear in these systems. And one of the things that we wanted to understand was, how do these flows depend on the kind of cells we have and the geometric parameters of the cells. So these are different cells with different lengths. This L stands for the length of the cell. And what you start to see is they form these colorful, this is color-coded microdomains. And these domains are nothing but nematic domains. And at the intersection of these three domains, you start to get topological defects. And depending upon the length of the cells and how fast they are growing, you start to see it controls the size of the domains. And so one essentially arrives at an emergence of an active length scale that arises due to competition between the orientational stiffness. So essentially, what is the resistance of the cell to undergo an orientation versus the extensile active stress. So these are essentially non-motile cells, so they don't move by itself, but whenever a cell divides, it pushes the neighboring cells. And the domain size then also depends on the inverse of the growth rate and positively correlates with the length of the cells. And it has consequences on how mechanical stresses develop in these colonies. So again, taking cues from liquid crystal science, it does turn out that if the cells are dividing along the long axis of this microdomain, then they are able to propagate their mechanical stresses more effectively, which is shown over here. If the cells are dividing perpendicular, then there is a buildup of stresses. And this creates these different flow patterns that you see over here on the left and the right. This is for the radial direction, and this is for the azimuthal direction. And the question that we wanted to ask was, these flows, can this momentum be transferred into the environmental fluid that encloses this system? And this was work by Joy and Garima, two postdocs in my lab. And what they found was very interesting. So depending upon how fast the cells are dividing, what the substrate properties are, they are able to specifically tune how much flows they generate. So the way we do these experiments is that we create these very well-defined confinements. We take these raw images, we segment them, then using a machine learning algorithm, we define the nematic domain texture, extract the topological defect, and we do this for different conditions. And so this is just a snapshot. You see these are specific bacterial colonies which are growing optimally at 37 degrees. So hence at 37 degrees, the colony grows up to a much larger size within the same time as the cells at 25 degrees, right? And when we compare these, you start to see there are different dynamics emerging, right? So this is the aerial speed. This is how fast the cells are growing. So at some point, they stop growing as a monolayer, and then they get into a multi-layer configuration. When we quantify all of these parameters, what we start to see is that there's a lot of variability in the system. And this variability occurs typically in the area up to which these colonies grow, the time the cells take before they go into this multi-layer configuration. And this is something we quantify with what is known as the Fano factor. It's the variance over the mean squared. And what you start to see is these are different parameters we measure: the critical time before the structural transition happens, the length at birth, the area, the single cell division time, and the aspect ratio. You start to see that the critical time at which the structural transition happens is typically has almost 2 orders of magnitude less noise compared to any other parameter. And this is in log scale. And we were wondering, why is this the case? And it all turned out to be, let's say, an interplay of different forces. I won't go into detail, but essentially what the cells do is they regulate their size as they are growing, because there is a buildup of stresses towards the center. And that has an effect on the growth rate. and that feeds back into essentially a suppression of noise in the critical transition. So finally, just a couple of slides. I'll just show you what implication it has on from a biological perspective. So what you see over here, this is the movement of cells as a colony is growing. So these are actual experimental data, and these colors indicate the strength of movement of the cells. And what we start to see over here, there are these small vortices which start to form. And if you see, for each vortex, there is an anti-vortex. So really these vortices are patchy. They form these pairs of vortices. And these pairs of vortices actually can be quantified. So this is the mean average vorticity, and this is the frequency. And finally, what they do is they enhance the transport attributes in the cells. So if you put in some tracer particles in that environment, if these tracer particles has a Brownian diffusion coefficient, which is indicated by dB, what we see over here is these flows, these vortices, give almost close to an order of magnitude enhancement in the diffusion, suggesting that these flows can help to homogenize different molecules in the system. And this we demonstrated at the time with some particle experiments. You start to see there is these particles are really getting torn apart. These are 2-micron beads held together by van der Waals forces. And these flows are strong enough to overcome these van der Waals forces. And this is something you capture through this coordination number. So without the bacterial colony, you have almost a steady coordination number of 4. And with the bacterial colonies underlying, you see there's a steady drop in the coordination number. So we use this data to then go a bit deeper to understand how, depending upon the fluid viscosity in the environment and the size of the microcargo, what kind of enhancement of the diffusion coefficient one expects. Okay, finally, the stresses that get developed in these cells and these colonies, these are in fact, there is a wave-like propagation of the stresses. So on the right-hand image, this is again a growing bacterial colony, this time simulations, where you see that there's this reddish hue coming up. And this is in fact, it's the magnitude of mechanical stresses which are emerging in the system. So there's still work in progress. And what I showed you till now were these low-order nematic orientational ordering in this bacterial system. But in fact, there are also high-order smectic-like positional ordering occurring in these bacterial systems. And this is something you can realize if you compare these systems. They look like layer-like stacks one expects in a smectic system. And in fact, when we look into the growth of the colonies and look at the smectic order, you see as the colony grows, the smectic order also keeps on growing. So with this, I would like to end my talk. I see there are at least 5 comments in the chat, so I will take them up and leave you with a quick summary of our, let's say, the key messages of this paper. And thank you once again for your time and the opportunity to interact with you today. Thank you very much.
Thank you so much, Professor Sengupta. So there is one question from Professor Shomnath Chatterjee in the chat box. If you can take this.
Yeah.
Sure.
So let me see, Professor Shomnath Chatterjee. I have a small question. How can you detect the nature of cells by EBSD techniques or confocal microscope? What is EBSD, sir?
Actually, EBSD is the electron beam scattering diffraction. It is actually it is with the SEM system.
Correct. Correct.
So as you were showing the topological view, that I thought that maybe this is due to the EBSD phase microscopy system.
Yes. Yes, indeed. Very nice question. Thank you, sir. So all of the images that I showed, these are done in phase contrast imaging or fluorescence imaging. And in fluorescence, we use also confocal fluorescence. So essentially, with an appropriate dye molecule, we typically able to do stacks and then get a three-dimensional reconstruction of the system. The other thing that we explore a lot is the intrinsic birefringence of the system. As I was saying, due to the liquid crystalline nature of these biological ensembles, if the wavelength of light is comparable to the structures that we are seeing, we can really leverage from the optical anisotropy of the system. Now, as far as the EBSD is concerned, we are doing this SEM imaging, but there is one, let's say, small, I would not say demerit, but we lose some information there because typically to do the SEM, we have to fix the cells and then we have to carry out the imaging. So in many of our studies, what we try to do is still keep the cells in their own native life state so that they're still doing their normal actions and functions while we are able to image them, right? But in some cases, we do SEM-based imaging, but those are mostly for static time point analysis.
Yeah, actually, sir, why I'm asking this question, when we are for the solid state means solid state pupils, we are using the EBSD for just seeing the domains, the different grain boundaries. So as you are using this contrast image, that's why I'm just asking that whether it is you are getting it from using the EBSD or not.
No. We actually got it only from phase contrast. So we use phase contrast and then we use image processing.
Yes.
And then we get these final features. But what you say could well be tried. It's just that taking live biological samples, preserving their structure and then doing SEM, we might lose some information there.
Yeah, I think so.
Yeah.
Thank you, sir.
But thanks for this very nice question.
Thank you, sir.
Thank you so much, sir. If someone has any other question, please ask our speaker here.
Yes,
Sure.
I have
Yes, sir.
I put one question in the chat box, even though this is a bit delayed.
Yeah.
Sir, it's really very nice, informative, and motivating talk actually. I was thinking that you have done a lot regarding to just identify such kind of microbial effect on different objects. One time I was also doing work some anisotropic diffusion, which takes place in the different mainly tissue architecture. And there we have seen that the diffusion anisotropic actual process. I have used this in microimaging in MRI.
Yes.
And there I've seen that it's very good to discriminate the diseased tissue from the normal tissue.
Absolutely. Yeah.
Because when tissue get damaged, then anisotropic part is lost actually. And in that case, the fluid flow becomes a little bit isotropic. That is one indication for the discrimination between diseased tissue and normal tissue. And there, we have done one work that to decide this based on some statistical hypothesis. How we'll judge that this is the with high degree of anisotropy and there is less anisotropy, and we were successful to discriminate this also. But problem is that we had used 4 millimeter sample size, and the machine was 500 megahertz.
So 4 millimeter in depth.
Yes.
The thickness.
4 millimeter thickness. 4
thickness.
Millimeter,
Yes.
4 millimeter.
So the light passes through 4 millimeter.
4 millimeter. Yeah.
Mm-hmm.
And there we have seen that it is really possible to discriminate such kind of an anisotropic diffusion process. To know about the hemorrhage on the tissue, it is possible, and based on some statistical hypothesis, obviously. But if we failed there, it should be supplemented by methodologies. Then it will be perfectly deterministic. So can you, Professor, comments on that? Which type of methodology can supplement this kind of experiment?
I
mean,
missed some, I think there was a bit of disconnection. The voice got disconnected. When you said supplemented by, what
by other kind of experimental methods, which can supplement this measurement also.
What I could, let's say on top of my head, indicate, you might already increase the signal to noise ratio if you take thinner samples. That is the, let's say, one of the first things. If we can go really to a millimeter or even sub-millimeter, then I think some of these effects can be more clearly captured. What happens, the thicker the sample, you have an integrated signal from all the different layers, right? So if we can really make slices thinner, then I think that would already help to increase the signal to noise ratio. In terms of having supplementary data, one of the, so if I may ask, how did you measure the diffusion coefficient, through tracers or?
Yeah, actually, we have taken basically the pixel data for different magnetic field and in different direction.
Okay. Okay.
So
yes,
in that way, one can determine the diffusion tensor.
Right.
If we equate that diffusion equation there, then in 3 dimension that D becomes the matrix of 3 dimension.
Correct.
So yeah, in this way just we determine the actually diffusion tensor.
So the magnetic field was fixed, or you had a frequency certain?
Sir, actually the main magnetic field was 11 Tesla, that is in Z direction.
Right.
And the radio frequency that has been applied, it has been actually given in different orientation. In this way, we have changed the mainly the direction of the magnetic field as well as also magnitude. So if we do this, then if we fit the equation exponential minus T dot D dot T, which actually basically nothing but the amount of magnetic field in radio frequency and in association with that one, the diffusion tensor matrix. So if we just fit this equation pixel wise, then we get the data of diffusion tensor from that pixel.
Absolutely.
So in this way, we can calculate the diffusion tensor, which gives us the anisotropic diffusion. Or we can calculate the what I say, the relative diffusion, relative anisotropy diffusion.
Correct.
That gives us the value.
Correct. Absolutely. 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 So what we of course in our lab we do not have access to 11 Tesla magnetic fields.
Okay.
What we do instead is we use optical traps, optical tweezers. And so what we can do is, if you have then, let's say, suitably sized beads. So there is this, it's now about 10 years or so, this field of active microreology. So you put in some beads and you optically trap these beads. So you can have an optical trap with multiple beams, and then you give a certain frequency. And you look at the relaxation of these beads, from which you can back calculate essentially the diffusion in different directions. But of course, I understand your sample size is also much larger, but typically what we do, because this is something we do on a microscope stage then, right? So optical is a very nice, I think, complementary technique. Then there is this more recent, it's now about 5 years old. It's called Brouillon microscopy, where it's a non-invasive microscopic technique. Just shining light through it, we can get, it does not give us the anisotropic component, but it gives us all the structural information within the cells. And from that, we can reconstruct the anisotropic.
Yes. That's good. Very nice, sir. Thank you. Very nice. Thank you very much.
Sir, I have one question. I mean, just I'd like to know that that for solid crystalline material, as we know that defect plays a crucial role in determining and controlling materials properties. And sometimes we deliberately introduce defect to control materials properties in various aspects. So as you are discussing about the liquid crystal, and you're talking about the topological defects. Now I would like to know that in which way defect manipulates the properties of liquid crystal. I mean, how it affects the properties of the liquid crystal, because I don't have much idea on that. If you could please elaborate a little bit.
Sure. So in liquid crystals, I think one nice way to visualize a defect is at the core of the defect, these long range correlations disappear. So the fluctuations, the molecular fluctuations are so high that the liquid crystal molecules do not know which way to orient. So essentially, you can consider for a liquid crystal, the core of the defect is essentially an isotropic phase. Okay. Now, when you have an isotropic phase, essentially everything which happens due to anisotropy disappears. So what it essentially means, if you are shining light, it will scatter the light a lot. So although the defect cores are typically few tens of nanometers or even like 10 sub-nanometer, but if you look at them between microscopes, even without polarizers, you will see them scattering light in micron size. So that is one thing. That's the optical manifestation or an optical impact. But think of it, you put in any other field in presence of defect. For example, if you put a hydrodynamic field, now compared to an anisotropic liquid crystal phase, the isotropic defect core has a different viscosity coefficient. So that means if you now impose a hydrodynamic field, the effective viscosity or the effective flow speed through the defect will be different compared to the surrounding ordered phase.
Yes.
Now, if you put in, let's say, an electric field, within the electric field, so depending upon if you now have from a point defect, if you have a line defect, which is also called a disclination, right? So if you have a disclination and you put an electric field, this disclination acts like a region in the system where no electric field gets transmitted. So people have actually leveraged it to put in some conducting nanobeads and use defects as short circuits in liquid crystals and so on. So effectively, you take any kind of field and you have to consider that the defect is an isotropic phase. So how would that field behave in an isotropic phase? Exactly that is what is happening if there are defects in the liquid crystal.
Thank you, sir. Thank you.
Okay, I see there are a couple of other questions. Okay, Professor Sharkar, Shobhik Ghosh. I've heard that quantum computation encounters a significant challenge known as decoherence, requiring the maintenance of low temperatures. However, today I came across an information suggesting that photosynthesis manages to establish coherent environment to enable quantum effects. Could you shed light on how a basic plant cell achieves this? Wow, yeah, this is a field in itself. So indeed, you are correct. If you just look at the Krebs cycle, which is the key cycle in maintaining photosynthesis, it's still debatable if actually quantum effects are in place. But there is a growing, let's say, line of research which is looking into the quantum aspects of photosynthesis. So unfortunately, I would not be able to comment on that because I'm not too sure if the quantum effects which people are alluding to in the more recent works, how robust they are, right? Because in biology, there's this whole field of also quantum biology. But yeah, I think it still needs some research before I could give a yes or no comment. So I will save that for some more time, but feel free to contact by email.
Thank you, sir. A few other questions are there still. Yeah.
Okay, yes, Shatarupa, can this bacterial world help in tissue? A very interesting question. Well, this is, I would not say what we see with our patient-derived cancer organoids, but we do see that they have an impact on organoids, right? And organoids is nothing but an ensemble of tissues. So the micromechanical forces that they impart on the extracellular matrix, that feeds back into the growth of these tumor cells. So that's very clear, right? The other thing, what people are also extensively looking into is how do these microbes or bacteria influence wound healing? So when you have a cut, and now because we try to always, let's say, disinfect and keep it free of bacteria, but there are also certain, let's say, microbes which have been indicated to enhance healing of wound. So what you are asking, I think people are already thinking about it, but it's a very pertinent question. So there's a lot of active research currently being carried out.
Yes, sir. Sir, I heard something like this, so that is why I was asking.
Yes, yes. It's a very well posed question. It's not not like just an imagination. So
thank you, sir.
Papia, yes, thank you so much. Is there a possibility to sense the cell through optical sensing? Is there any photonic crystal structure in colony of cells? Very good question, Papia. We are working a lot on this field. In fact, many bacterial systems, I would not say the one that I was showing you today, but we have a whole line of research where bacteria, also algae, they are known to precipitate various forms of minerals like calcium carbonate, like calcite, aragonite, and they do so with different morphological features. And some of these morphological features are actually of the wavelength of light. And of course, then they have very interesting photonic properties. There is this other field of research, it's called, when you look at photosynthetic algae, so these are green algae which also produce oxygen. The algae, there's a class of algae which cannot swim. They are called diatoms. And diatoms typically have what is known as a frustule, F-R-U-S-T-U-L-E. So if you just search it on Google, you will see these frustules have almost like metamaterial-like architecture. So these are extremely well tailored by nature mineral structures which grow on microorganisms. And people are trying to mimic that in lab for photonic applications. But in nature, it's already there and in vast amounts and diversity.
Thank you so much, sir. Actually, I was thinking if there is a possibility of optical sensing of this kind of cells.
Yeah.
But it's difficult because laboratory arrangements should be so advanced to sense.
On one hand, yes, but on the other hand, as I said, to get a basic thing started, it should not be that difficult because technically, if you have access to just a polarized microscope or a microscope where you can put in two polarization films, you can already start to quantify many different features, right? So I'll just quickly share one thing so that you get to see what I meant. It's from another talk I gave some time back. So sorry about the outdated slide. Let me know if you see the screen.
Yes, yes.
So this is actually aquatic microbe. But look at these very intricate textures, which are essentially silicon deposited, so silicates, which make up the cell wall. And so when you shine light, it really has a lot of interesting optical properties, including like you can get whispering gallery modes and so on, right? And so these kind of organisms are plenty in nature. And if you see, if you just shine light, and you will get very interesting birefringent properties. So also theoretically, then you can really create some interesting research questions, right? How does the overall morphology and distribution of these structures would allow photonic properties to emerge, right? Yeah, so you see these are SEM images of the cells, and if you look at the scale bar, so these are typically sub-micron scale features. So essentially very suited for photonic purposes. So there's a huge field of research happening.
Yes, sir. Thank you so much.
Okay, about photonic crystal structure column. Okay, so this looks similar to Papia's question. Atmaja. Big role of minerals. Absolutely. There's a huge role of, so there's this huge field of, it's called biomineralization. You have to just look it up. It's a huge field of research. It's an active field of research. There's a lot happening. It's called biomineralization. Yes, Shobhik, indeed, we can harness some of these. But you know what the thing with biology is, as I was trying to explain, biology has its own variability in the system. So to take something really biological and get a computer, like with a very deterministic and robust functionality, one has to bridge that gap, right? So there is still a bit of gap between what biology offers. Biology suffers, well, biologists would disagree with it. Biology has a lot of variability, and variability is there because evolution requires variability to be there. Otherwise, species will just get extinct. But on the other hand, all our computing and physical processes require robustness and precision. So these two are a bit conundrum one has to bridge. Okay.
Okay. Thank you so much, sir.
No, thank you. Thanks a lot. I really enjoyed this interaction. And I should admit, it's not that often we have such an open chat after the talk, because, once the talk is done, I think people are eager to also move on. But I think today I really enjoyed the discussion.
Much more lively if you would have come here. So next time we are expecting you to please visit our university.
Absolutely. I would, as I said, it really turned out a bit, we were all caught a bit unprepared with this development yesterday. And I had a chat with Maharshi this morning, and I said, yeah, next time I land in Kolkata, maybe I first come to Adamas before I head to my family.
Sure. It would be really nice. So thank you so much.
Thank you very much. And
our faculty members as well as students, they have enjoyed this talk very much, and really thank you so much.
Thank you. And yeah, if there is anything, if you have any other follow-up question, comments,
yes. So I think I'll
me
share your email ID with all my students, and I'm sure they will be able to interact, even if they have some question later.
Absolutely. Absolutely.
Okay. Thank you so much.
Thank
and
you.
I would also like to thank all the participants for joining. Thank you.
Bye-bye.
Bye.